time in Thailand, Bangkok, and I will try to share our experience in Turkey, how we try to design the DNA technology in clinical medicine. And this morning I will try to talk with you about how DNA changed our vision in medicine. So it's a very active program actually, because it is an integration of new biotechnology into medicine. So everything starts with DNA, you know, and it's not a very long history, but we know that everything is in DNA, and we carry the information to next generations from it. And then information in the DNA is translated to proteins which function in the body and regulate all the human organism. And clearly the most important thing is we carry all this information to next generation. So maybe it's the life, unique life in a person, and it is carried through all the generations. So when we look at the story, maybe if we can have Mendel like 18th, the story is not too long. And when they discovered the DNA with Watson and his friends, it was like 50 years ago. So after that, it's a huge, very fast development, and we come to embryo screening, embryo technologies, cloning, and finally, after 2000, we aim to sequence and learn all about the human genome. It was a huge project, and the first member who was sequenced was also invested this project, Craig Venter, and it was like this year we can get the first information about this project. It aimed at a very big, one of the dreams of the humankind, to learn about human DNA, what we carry, how we carry it, and what's the meaning of this DNA. And there were main topics what we aim to have after this project. And when we group them, we can just have the first aim is to map the human genome. So the first aim is to know the diseases, genetic diseases, carried on the DNA, and have the physical maps and make it easier to diagnose the diseases on the DNA. So almost this is trying to be completed, and of course with the unknown parts. And the second is maybe the most important of the result of this project is have the new DNA technologies, because this makes us to understand DNA and put it in our daily medical life with these technologies. First, they were very expensive and not very easy to apply them in daily medicine, but now with the new techniques, microarrays and new sequencer technologies, we can use them in our daily medical life. And sequencing human genome, we understood it is not very easy. And it is not very easy to sequence all of them at one time. So now the technology comes to sequence part of it and then complete the sequence with informatics. So we need to have a very expanded programs, informatic programs for the sequencing. And we come to a point that sequencing of all the genome is part of something, but we can sequence the meaningful or the clinical parts of it, and we come to a point that we can do exome sequencing. So as far we are learning, we are trying to get the new technologies for our daily medicine. And what becomes most important is to get all this data. To get all this data, to keep them, to project them into new studies, and is a big work. So even now we can get the very advanced labs. It is very difficult to get the informatic basis to all the units. So what we do usually we do the technology sequencing or DNA studies and then try to gather them in the big informatic centers and get the data from that. So this is very important, and this is cloning in silico, meaning that it's now growing, maybe the most important thing to get this information from DNA. And by these studies, we have to find easier ways and to make us faster and get the results. So we use comparative genomics, which means that we can just learn from other species, which is easier, and compare it with our homologs in human. So we come to a point that the most functions, the pathways, as we know, are similar. So there may be a genetic relation between the species for that basic metabolic genetic pathways also. So functional genomics is now the point where we started for the function of DNA, expression of the genes, and this is medicine, because we are more interested in the function of DNA if the genes are working in a person in the right time, or because of anything happening in the DNA, or during this pathway, there is an unfunctioning pathway which resulted with a disease. So there is a problem which we have to deal with. So this is the expression of the genome, and we come to a point that it is post-genomic genetics, which means that we are more related with the clinical side, functional side of the DNA. So one of the most, how we do this in the clinics, of course, the microarrays are very easier to use for us now, especially for these expression models. What we do is usually, if we have any kind of sample of DNA, we can compare it with the unique wild type, which we know is the normal, or at least we know the sequence of this DNA. And when we compare our sample with this DNA on a chip, we can get the parts which are amplified or deleted. So meaning that there may be something wrong with this sample, and then compare these results with the data we had before. So all these technologies, when we combine, it is not very difficult today, and it's usually doesn't take too much time to go through the samples of DNA. So in medicine, it means a person or a patient, and get the knowledge of his functioning DNA. So when we estimate, we learn about that person and that patient, about the disease. Can we touch the DNA, or can we modulate the disease, the pathology in the DNA, or functioning of the proteins? So is it good to play, or do we have the right to play with DNA? Of course, the most important thing is another big lecture maybe, but the most important thing in the subject is to think about the structure of DNA. We may have somatic cells, somatic DNA. It is related only with that patient, with that person. But when we talk about germline, it means that next generation. So it is ethically, not only technically, but there are huge considerations in ethical ways in biotechnology, because it is related with next generation. So if we touch DNA, it means that there's always a question mark what we are doing for the next generation. So what is this personalized medicine? How we can combine all this information, which is invested a lot, and how we can get the results and use it for a unique person. And is it real? It really gives us something to have a new vision for medicine. So what is planned, or what is going on in the new clinics is that we have all the data about a patient admitted to the hospital, the radiographic information and the blood work, but now the doctors may have the patient's molecular tests and screens on his hand immediately when he sees the patient. So now he has the chance that seeing all the patient's pathways, DNA structure, and the functioning parts, and unfunctional may be related with the disease. Now she's admitted to the hospital, and the potential risks which he can be related later on. And of course this is important not only to diagnose or to follow the patient, but for the treatment options, which I will talk later, is very important, because then he can just, he will know that which therapies she will be getting more benefits. So now the doctors may have a chance to see deeply inside one unique patient, and it is not a statistical result, or he has to consider all the statistic results for that patient, but now he has unique information about that person. So in modern medicine, what we are trying to deal with is we use all this biotechnology to take care of an individual patient using the genetic technology, and because of the information shared with the patient, maybe the most important thing in the modern medicine is to involve the patient, involve the person who admitted to the hospital, talking with the doctor to decide about himself. Because before medicine, usually the doctors were the God, and they decide what is going to happen to that patient, because there was not too much information to be shared with that patient. But now we can have the consultation, and the patient gets all that information. So he will be involved in the decisions from now on in medicine. So maybe this is the basic new vision in medicine, modern medicine today. So when we do this, when we use this personalized medicine, is it only for the adults, or after birth maybe with the family permissions, or we can do the prenatal work, or even now preconception, and we can use these technologies in the embryo. So we can do personalized medicine to all these unique living organisms, and we can use preventive medicine with preventive genetic diagnosis. Of course, always considering about the ethical points. So now it comes to a point that when a baby born, with the screenings we are doing now in medicine, but also make them rich information for that baby with molecular screenings. So with the sample from a baby, we can know all through her life the risks, the advantages and disadvantages of her to carry all through his life, and keep them in a chip. So this chip is unique for her and can be used all the medical society in the world internationally, and it shows only for this baby, for her DNA. So what is this unique in the in one person in the DNA? Usually we have the the simplest way is our functioning proteins, the amino acids, are gathered with 3 nucleotides in the DNA. So if there is a change even in one point, we call it single nucleotide change, polymorphism. Sometimes it doesn't mean anything, it doesn't change the sequence, the protein. But sometimes it changes the proteins and makes the functioning part a little bit changed. So if this is kept normal in the body, we call it a polymorphism, very general. So it means that this person has another type which functions normally, but different, like 10% of the population in the world. So it is a normal function, but can be another type of a normal version. But sometimes this changes doesn't compete with the normal function of the protein, so there is the disease now. So it is very important, even in one nucleotide, in one point of DNA, if it is a polymorphism or it is a mutation resulted with a disease. So this is not only the medicine medicine point, but when we group the people, can that be real big point to distinguish people with the DNAs? Is it really very significant and specific? When we go through the DNA with 10 markers, 10 points on the DNA, we can just gather like 8 million people grouped in the population. When we go through 25 points on the DNA, there are only like 250 people in these groups. And when we look only 33 points on DNA, there is only one person in whole society, in all the world. So it is very significant, and it is the real signature of a person when we look at this information on the DNA. And in personalized medicine, we usually, the old medicine, we usually treat and follow our patients with trial and error methods, the statistics. So when we go to the modern side after the DNA, we can say right treatment for the right person at the right time very significantly. And also the treatment, we know that it is not so much significant, and usually there is like 10% of people who doesn't benefit the regular treatments. And this is not maybe the dangerous side, but the dangerous side is almost like 10% of these treatments are toxic or have adverse reactions for that people. So it is not a modern medicine anymore for the 21st century to go on with these classical methods. So what we have now? We have now some information from DNA which helps us to diagnose an early, it may be an early detection of the risks for many kind of diseases or pathologies. So we can look through your collagen genes and we can estimate about your bone function, bone biology during your life and guess about your osteoporosis. And metabolic pathways are, very important in aging, in other diseases, so we can get many information from your DNAs. The most important thing is to do this test with the licensed information background. Because in the internet, when you open, there are millions of genetic tests which can tell you how long you will live, which diseases you will have, but usually they are not licensed and they don't have the collaboration or meta-analysis results of the test. So we have to find out the real informative tests and which can gather all the information after this DNA test and get you a personalized report. Usually for the reporting, what we do it after this test, we do is significant for that person with the daily medical information. We combine the DNA report with daily results, clinical evaluation of that patient, blood work, and then evaluate a personal report of what we advise, how we will follow the patient. So what we do in the clinic is, we call it personalized health management, or you can call a personal or preventive medicine clinic. We do the counseling first, the examination. What does that person, usually the people without the disease come admit to these clinics. So what is this person having the family background and the daily blood analysis, everything to be checked in this first consultation. Then we do the DNA analysis and prepare the personal report combining all of this. And after this report, there is a huge team. It is not one doctor anymore, so it is a team work. The nutritionists, the dietitian people, the psychologists, and the doctors, cardiologists or oncologists included in this team for that person. And usually headed by a family doctor or a preventive medicine doctor who organizes all of this for that person. So from now on, this person is excluded from the statistical medicine and come to a point of personalized information and personalized medicine follow-up during his whole life. And genetic counseling is very important because it should go through all his background and this person should know the results will be related with the next generation of her family also. And genetic testing is easy. You know, we do it with blood or even the smear from buccal part. And the results, technical results are like that. So many doctors now in the clinics get used to these reports. So we have the genes which are related with our clinical functions, and this is the results from two alleles from mother and father side, and we can say the result of the genetic test as there is a homology or heterology or what can be the result of this change in the DNA. So we can tell a lot, and at the end it comes to patient like that. He may have good genes, he may have bad genes, and he may have a daily or medical life, which can modify his benefits or advantages and disadvantages he is carrying from birth. So we tell him that medicine is here. So we can modulate these benefits or disadvantages and change them to good points or prevent his risks and can result with a longer and healthier life. So it is polymorphisms we carry. We combine them with environmental factors, meaning medicine for us here, and we can have the follow-up. And this is the result because the main question is, this is the first generation in the world who is applied to this genomic-based medicine. So we really do not have big information or a scientific evidence that this follow-up, including DNA technologies and preventive medicine, really longers and gives high quality of life to a person. So the results may come after this generation, but what we know is it really affects the environmental changes. It means medicine now changes the genetic background. These are twins which are grouped different lifestyles. She's in the church and she's working in the farm. After 60 years later, when they come together, there's a huge difference. This picture maybe all of you know. So the estimation and the thesis, the experiments show that the gene information in medicine and to go on with this medical information for prevention really will affect the life quality and the time period of humankind. So it is not only the person by person change with the aging or gene structure, but also our organs can be aged differently in a body. Your heart can have a different lifestyle model from your liver, for example. So what we do is we use this technology in adults after birth, in fetus or in embryo. We come to a prevention point with embryo because we have now pre-implantation genetic diagnosis. Ethically, we can only use this method in fetal diseases like thalassemia or Down syndrome. So we got one cell from the embryo, and in our labs, we get this nucleus from that one cell and get the DNA information test from this cell. And detect if there's chromosomal abnormality, like that clearly, or thalassemia like these embryos. This couple has like how many? 10 embryos, and we can select only the normal without thalassemia embryos like this two and transfer it. Although we can see the chromosomal information by fish, so we can know it is not carrying down abnormal chromosomes also. So this is
1 minute.
Okay. So this is the technology we use, and in conclusion, yes, we can do the prevention, but what about the future? Is there a magical way which we can use to cure if there is happen a pathology? Stem cells. Is there a magical drug that we give the patient, prepare the drug, give it to the patient, and then have happy results? So is there possibility? possible. So we have the stem cells now. So we come to a point that we can get the cells from the body, and these cells, when we use them with official way, and we now have personalized medicine, preventive medicine, and we come to a point that it is regenerative medicine. So when we go through the cells, which we can use in the clinic, which I will do my second speech and show you some results, today we can get enough number of stem cells from a person, fat or bone marrow or other parts, and use them in the therapies which are studied before in like this labs, and this is the center we have now. It is like an IVF center. We have the labs here and the surgery rooms here. We get the samples and modify the cells here. Not modify, but get the cells. And then we can create a cartilage for a person. We can use these machines in the surgery room and cure that bones in 4 weeks only and get rid of a 3-year wound from a 76-year-old patient, burns or cosmetic. So in the second speech, I will try to get you more information about regenerative medicine. And this is our clinic in the university. Thank you very much for listening to me.
Thank you very much, Dr. Excellent. Is there any question from the audience? If not, thank you very much. Oh, sorry, you had, sorry, go ahead. I didn't see you.
Yes. Thank you very much for this. I'm slightly concerned with your emphasis on genetics being personalized medicine, because we've heard all the way through this conference that when we talk about environment, nature, nurture, that many of the lifestyle diseases we're talking about, which afflict this world on pandemic status, are multi-genetic. Many of those genes can be modified by behavior. Wouldn't you like to see the two brought together, or maybe you do?
Yes, I have to go faster, but this is what we are doing. Environmental means in some clinics, of course, it is lifestyle changes. But in medicine, of course, it's an environmental modification for us. So when we see a patient, it is we get the DNA and blood work, the clinical status of that patient in that day, and try to get all the classical medical follow-up and treatments for that patient. So we combine it like environmental methods. So we send the patient to a life change or lifestyle control clinics. They are outdoor clinics like here, but also the follow-up in medical clinic is personalized. We do the checkups for him if she has a breast cancer risk, for example, and she is already 30 years old. We take her out of that statistical group, which the checkup usually starts after 50 years old or 40 years old, and we start her checkup when she's 30, and we try to regulate her bone structure because we cannot maybe give her hormone therapy. So this is all have to be controlled for that person according to her DNA and clinical status of that day. So medical follow-up is a kind of environmental modification for us also. We try to combine that information.
Thank you.